So I'm giving 3 kind of shorter 15-minute talks on processing, immunogenicity prediction, and some of the analysis tools. So for the processing tools, is the microphone on? Yeah? No, it's not? Okay. Okay, better. Okay, so for the processing tools, the motivation is that for a peptide to be recognized by a T cell, that doesn't depend just on its ability to bind to MHC. So we have two types of processing tools integrated into the IEDB. Now I'll go through the interface and prediction outputs, and also talk about the performance and caveats. And I'll do the same thing for a recently developed immunogenicity tool focusing on MHC class 1 immunogenicity. So this is my incredible graphic artist skills. As you see, it's a mouse, but I have to label it clearly so that you recognize that. And this is a virus. What happens when a virus infects a mouse? So it goes inside the mouse, it infects certain cells, starts replicating, and from those cells, and essentially replicating throughout the body. And what happens inside the cell is the interesting bit for us. The virus, and actually every protein in the cell, gets cleaved into peptide fragments, and some of these peptide fragments end up presented by MHC class 1 molecules on the cell surface. And then you have T cells that can recognize these peptide MHC complexes, triggering an immune response, resulting in cytotoxicity, cytokine release, or proliferation of the T cells to actually form subsequent memory populations that then can rapidly clear viruses if they reoccur. So again, the T cell epitope mapping problem then is to define which peptides within a virus would actually be targets of such an immune response, which are the T cell epitopes. And essentially, for the processing part, the question is which peptides actually end up here on the cell surface. Obviously, they have to bind MHC, but other factors play a role. So essentially, this is what happens inside the cell. Every protein actually is subject to degradation by the proteasome, and you have these peptide cleavage products that are generated as part of regular turnover. What happens normally with these peptides is that they are cleaved into single amino acids, and they are used to resynthesize new proteins. So you actually, cells are not a static environment. There's a continuous turnover of all the proteins inside the cell, and in this process, these peptides get generated. And so essentially, on that, there has been this piggyback development of the immune mechanism, where this transporter associated with antigen presentation tab grabs some of these peptides, transports them into the AR, where they can get cleaved further, and then bind to MHC class 1 molecules. And these MHC class 1 molecules, if stable, get transported onto the cell surface, where they can be then scanned by T cells. So this is the whole life cycle, if you want, from epitope contained in a protein to what it ends up in the surface. And as you see, there's multiple steps involved in this, and each of these steps has actually been characterized experimentally to some degree. And we have quantified how important they are. So essentially, about 15% of all possible peptides are generated and are available for MHC loading in the ER. Within these, maybe 2.5% at best are capable of binding to MHC. If you have a peptide MHC complex on the cell surface, for about half of those will you have an immune response. So if you're essentially trying to see which peptide in here is going to end up here, the dominant factor is to understand what's causing MHC binding, because that's the bottleneck essentially of this process. But clearly there's other steps involved as well. So there's two ways people have tried to essentially incorporate these factors in predicting epitopes. One is a combined predictor, where essentially these individual processes of proteasomal cleavage and tap transport are characterized based on in vitro systems, and then that's put together essentially together with the MHC binding predictions, and then you have like a combined predictor. Then in addition, there's two predictors, NetShot, NetCTL, that are is trained actually on the ligands that are ultimately coming out of the processing pathway. NetShop actually specifically deals with the cleavage of the proteasomal cleavage, and then CTL is an overall predictor combining net MHC and NetShop. And then there's a specific natural eluted peptide predictor, MHC-NP, that was actually developed by a Canadian group, and that's hosted at the IEDB. That is on one of the epitope prediction contests. And then, okay, the immunogenicity predictor I'll talk about later. So starting with the combined predictor here, when you're on the T-cell epitope prediction tool page, you see here the link to the combined predictor, and essentially it looks at which should at this point be familiar, the typical sequence interface, the typical MHC allele selection interface, but then in addition you have these boxes here for proteasome and TAP. So for the proteasome, you can select two types. There's an immunoproteasome and the constitutive proteasome. The proteasome is responsible for creating the C-terminal end of most of the peptides that end up being presented. And essentially what this tool does, it looks for a sequence motif that is compatible to such cleavages at the C-terminus of the peptide. That's the proteasome. Then for TAP, that's the lower part here. TAP takes the peptides, as I showed you, that are cleaved by the proteasome and can transport them into the ER. And the thing is, though, that the peptide that TAP transports to the ER may not be the one that ends up bound to the MHC, because in the ER, peptides get trimmed further from the end terminus. So essentially, TAP has to be able to transport the C-terminus, but the N-terminus might be further modified. This tool, again, works based on predicted TAP transport efficiency, which was based on binding assays, and then because you have this effect that the ultimate ligand might well be trimmed from this N-terminus, there's a statistical model that is described by these two parameters that essentially asks for what is the effective average precursor length and how often do these things get trimmed. So if you do want to mess with those parameters, read the paper and then modify them. Otherwise, I would just leave them as is. This was the optimal setting for essentially predicting epitopes. So when you run these predictions, you're now getting additional columns here for the proteasome, TAP, MHC, processing, and total score. So the proteasome and TAP score are just these individual scores for the extended peptide. MHC is a transformation of it's like a minus log 10 IC50, just to get them to similar signs. And then there's like the sum of the proteasome and TAP and the total score, which is the sum of all three. So essentially you're ending up with a score again based on which you can take the top percent peptide. So when we evaluated these predictions, what we found is that when predicting eluded peptides, incorporating proteasomal cleavage and TAP transport gives better results than MHC binding alone. However, when we are looking for epitopes, we have not found that this actually leads to a drastic improvement. And some of the issues are that the data that we have available for human proteasome and TAP, it's only for human. Secondly, it's not that much. It hasn't been growing, actually. Not many people are doing these experiments. Versus the MHC binding, we're getting more and more data, and it's getting better and better. Also, this kind of seemingly contradiction that it works for eluded peptides versus doesn't work so well for epitopes might be because when you look at the eluded peptides, especially the ones that we use here for the benchmarking, they tend to be the best possible ligands, as in they're present in very, very high amounts on the cell surface, and so for them, everything might be working perfect, TAP, transport, proteasome, and MHC binding. But for epitopes, it's known that you just need very, very few peptides on the cell surface and you get an immune response, so that it might be possible to overcome suboptimal processing. So in practice, what we've been doing is we stuck to MHC binding predictions, also because it's just simpler to work with. But if you have a problem where you have say, a bacteria and scan that for class 1 specific epitopes, and you get tens of thousands of peptides which you can't possibly synthesize, This is a very rational, good way of cutting down and focusing on the ones that are more likely to be active. So next, I want to talk a bit about, this is the second implementation here. These are the neural network-based predictions of proteasomal cleavage and T-cell epitopes. This is essentially a suite of tools that build on each other. So NetShop is the proteasomal cleavage prediction. NetCTL combines NetShop with tab transport, which is the same as the previous one I showed you, and NetMHC. NetCTL PAN does the same, but combines NetMHC PAN versus NetMHC. So here, if you want, the key difference between these and the other algorithms is the use of NetShop versus the other proteasomal prediction model. So the effect is the same. So the interface is the same. You enter your sequences. You have the choice here between the proteasomal cleavage predictions and the one we would recommend to work with is the C3.0, which is essentially derived from MHC ligands and just looking for what does the motif look like at the C-terminal site of those residues. So while this might not be a true proteasomal predictor, it does perform better than the 20S 3.0 version of the proteasomal cleavage predictor, which is trained on in vitro digests. These are a bunch of references for this, mainly for if you actually want to read up in this in detail. The last processing prediction tool is the MHC-NP. This is directly trained on eluded peptide data. And there's a limited number of alleles available for this because this is essentially the data that was trained on. Again, the credits for this tool go to this Canadian group here. And essentially, yeah, you can use it like all the other tools and essentially treat it like, because it just essentially is rather than using binding data, dilution data, and you get essentially a prediction that should be more suited towards sales ligands. Finally, the MHC immunogenicity predictor. What we did here was we assembled large-scale. This is an example of data mining from the IEDB, straightforward data mining IEDB. We look at all data peptides that are recognized by MHC in the context of MHC class 1 restriction, or that are not recognized but bind with similar affinity. So obviously, if it doesn't bind MHC, it's boring. It won't be recognized. But we took epitopes, known epitopes with strong immune reactivity, and then we make a reference set with peptides with no immune reactivity, but that are binding at the same affinity level, actually using a paired approach to the ones that did have immune reactivity. And then we asked, is there an enrichment in the peptides that are recognized by T cells for certain residues? And what you see here is in black, peptides that are recognized, the residue frequency in peptides that are immunogenic versus white, the residue frequency where they are not. And so for tryptophan, phenylalanine, isoleucine, you have an overrepresentation of these residues in peptides that are actually triggering an immune response. And for both tryptophan and phenylalanine, this is actually been described previously also in purely experimental studies, where it's essentially found that long side chains in these residues are probably more likely to interfere with the TCR-MHC complex formation and thereby trigger TCR signaling. In context, some other residues like serine, methionine, lysine show the reverse pattern. They are less frequently found in immunogenic peptides. So we can take these scores now and essentially then see, okay, give me a peptide, then I can just go through these residues and see that this is overall the residue combination that is more likely to give us an immune response or not. This is the tool that does this. You can essentially just put your sequences in, and we are masking here, that's important, the MHC anchor position. So one of the problems in this evaluation is, again, like MHC binding, because it's such a crucial factor for the peptide to be recognized, you want to avoid it. So we're ignoring residues that are likely in direct T-cell contact in this immunogenicity scoring. Also, those residues will actually not be pointing towards the TCR. So they are less likely to actually be causing immunogenicity. So the tool is validated only for 9 myths. You run this, and you're getting a bunch of scores here. And I can show you in the next slide how to interpret these scores. Essentially, well, a high score means that the peptide is more likely to be immunogenic. And this is, we did actually in the paper, I think 4 different validation data sets, because we really want to be sure that we're not picking up some kind of statistical artifacts, which is easy in these things. And this was like another, like the 4th and final and completely independent data set, where we looked for peptides that are epitopes and non-epitopes in a series of human, so dengue epitopes, I believe, dengue class 1 epitopes. And essentially, when we plot now the scores that we got from this tool for these peptides, you see that I think this is like a total of 21 peptides or so, and 3 have a score less than 0, and 18 have a score above 0. Versus for the non-epitopes, it's pretty much half and half. So by asking for peptides that have a score greater than 0, you're losing a few actual epitopes, but you're cutting your set of peptides to synthesize it. So yes?
It's interesting, those amino acids in relatively hydrophobic are not going to be putting our T cells on. So you're finding them in a T cell response.
Yeah.
These aren't what's considered to be immunogenic amino acids in the T cell.
Right. So this is definitely a very much MHC class 1 specific immunogenicity prediction. Yes. Yeah, and I was actually surprised. I was definitely surprised by seeing lysines here being non-immunogenic. That's kind of a surprise to me. Yeah, but they shouldn't be. So I mean, we are looking only at the positions where they should be exposed, and I mean, who knows?
That's contrary to what you do.
Yeah.
What's the red bar? Red bar?
Okay, red bar is the frequency of these residues in overall. So some residues are underrepresented, like the lysine, because they tend to be poor MHC binders in general. So others are, yeah. So this is the kind of the confusing. I mean, so this is as I said, right? Like we're comparing peptides that are binders, non-immunogenic, and binders and immunogenic, right? So that's the bars. And then red is the frequency of these residues in the overall dengue virus that was scanned. No, this is not positional. Yeah. So we would like to do a positional. Yeah. If we could, if we had enough data, we would like to say residue 2 or residue 3 might be there, lysine is great or so. But this is, I personally like to stick with the simplest model first. Presumably the different positions should be somewhat similar in what they like or don't like, immunogenic or non-immunogenic. The moment you start divvying it up, and then you end up with very low numbers, it is very easy to get some weird kind of artifacts because the data set gets too small. So that was the immunogenicity tool. The last one is the section here of the analysis tools. And as I mentioned at the start, these analysis tools are meant for adding value to epitope data sets that you might have yourself or have generated by querying the IEDB. I gave you the example of swine flu. So these are the tools I'll go through. I'll talk about population coverage, which predicts essentially how a set of epitopes, what degree of reactivity would have with a given a known HLA restriction of these epitopes in different populations. I'll talk about the epitope conservancy analysis, which I had mentioned before briefly, and the epitope cluster analysis. Julia is later going to talk about homology mapping, I hope. Yeah, yes, not, but. So population coverage. As I said, the tool is meant to calculate the fraction of individuals predicted to respond to a given set of epitopes with known MHC restriction. So as you will probably know, different HLA molecules have vastly different frequencies in different populations. Actually, HLA are typically used to track movements of different populations over time in migration patterns, because it's such a highly diverse molecule, but obviously it's inherited and you can track it well. We do this here based on HLA genotypic frequencies and assuming non-linkage disequilibrium between HLA loci. And the ones we are using are coming at this point from HLA-frequencies.net, which previously used DBMHC, but now we switched to those because they actually have a much broader data set available. So for the example query here we're doing, so we have 11 MHC class 2 molecules that have different MHC binding patterns, and the question is, what is the population coverage in different North African populations? So you use this tool by adding here the population that you're interested in, and adding down here the epitopes. And actually, you can just add anything you want in here. This is just essentially a marker of what the epitopes can be called. But then, more importantly, what are the HLA restrictions that these peptides are known to be recognized by? And this could be based on a prediction that the analysis you done before, or the real data that you might have at hand. One important thing to note is that, as you see here, North Africa specifically, there's no mention of Egypt, for example, right? So we are relying on the HLA frequencies that are available from the public databases. You should definitely check back in when you do these kind of studies, what is this actually based on. So I mean, we show it here what this is based on. But now essentially here, from these populations and the population average will be calculated. This is the kind of summary output that you're getting. You have the different populations that were selected, and then you have coverage, average hit, and PC90 calculated. And then for each of the populations, you can click on them and get one of these links, and this explains all of these in more detail. So this is kind of the graph. So there's 22 total epitopes in the set I gave. And then here is essentially the predicted percent of individuals in that specific population that would recognize none of those, or 2, or 8, 9, 10, 11, 12, and so on, until like some guy actually recognized 21. So you tend to get these kind of complex patterns of this, and essentially the summary metrics we've devised are coverage, which is essentially how many people will recognize at least one epitopes in the set that you gave. Next one is the average number of hits. So when you just essentially say, okay, we have whatever 2% here that recognize 21, and then you have 0.5% that recognize 20, etc. You can add those all up, and then you get an average of about 6 epitopes per donor. But then actually in this case, both of those numbers are kind of, well, definitely this one is kind of misleading because you do have a substantial fraction of people that are predicted to not recognize any of these. So the PC90 is like at what is essentially the 90% confidence threshold for the minimum number of epitopes recognized with 90% confidence, and that's like half an epitope in this specific set. It isn't research, I think it's half an epitope. Yeah, so yeah, these plots are somewhat complex, but they can be useful, yeah, for essentially seeing if the sets of epitopes that you have are likely to be recognized not only in the population that you derive them in, but also in another one. In terms of conservancy analysis, we went through this before. This calculates the degree of conservancy of an epitope within a given protein sequence set at different degrees of sequence identity. And we define conservation as the fraction of protein sequences containing the epitope at a given identity level. So I showed you this example before. This is from the swine flu set, where we queried for epitopes in swine flu, entered them here, and then here added, queried for epitopes from seasonal influenza strains from past years, and then put them, asked which ones of those are conserved in a set of, I believe it was 10 different H1N1 sequences from 2009. And this is the output that I want to explain in a little more detail. So for each epitope here, you have the epitope, and then in this case, it is not found at a 100% identity level. Sorry, I should have shown this in the last slide. No, I should not. not found at this identity level at all in any of the sequences. But here, this one, for example, this peptide is found in 55 of the 439 sequences. And as we put in all the different, each HLA, each influenza virus has 10 or 11 proteins. So actually, this one is found in every single one of the strains in its respective protein. So here are the hits, and that's where you can see it. Essentially, this exact epitope is found in these different, you see this is a strain from Netherlands, China, USA, etc. So this would be was an example of a beta epitope that's actually conserved completely. You can then change these thresholds and look for different. This was just made at 100% identity. You can obviously change that and relax these criteria somewhat. For the cluster analysis, this is, as I said, meant for if you have sets of epitopes that have limited variability, and you essentially want to define clusters as groups of sequences which have a sequence similarity greater than the minimum sequence identity threshold identified. Okay, so you have a set of sequences, and they are variants of each other, and you want to group them together in sets of sequences that have that are similar to each other. This is how the tool looks like. This is, I believe, I shouldn't say anything wrong, but the sets of beta epitopes we actually queried from the immune epitope database. You put them in here, and then essentially choose what do you want to consider to be a similar peptide. In this case, we choose 80% sequence identity between two peptides. When you submit it, then this is what you're getting. So there's one peptide up top here that has no sequence similarity to anything. And then here you have a big cluster. You can see that this same peptide is contained here in multiple variants of other peptides. So yeah, with that was the quick run through the analysis tools. Yeah, any questions now? That's good. Or we also close to the break, right? Any questions right now? Oh.